Maybe give them like 1 more minute. It's the Friday effect, I guess. Okay. Hello, everyone. I just turned on my video to just say hi, but I'm going to turn it off as I proceed through the lecture. I hope everyone is doing well, and I'm going to start sharing my screen now. Okay, can everyone see the screen?
Yes.
Okay, so I remember this is where we stopped last time. And the question that I put in, before we left is if a molecule such as NKX 2.5 is involved in the primary looping process, and what sort of phenotype do we expect when the transcription factor is depleted from the system, that is the mouse embryo. Did anybody had a chance to look it up, or shall I just show you some cool pictures? Anybody wants to take a wild guess as to what might happen if the whole looping process is interrupted in the beginning? Okay, so I'm just going to move on. And so this is what the mutants will look like if there is a constitutive depletion of this factor. You can see that the embryos are way too small, and it's definitely lethal. However, some people think that it's primarily due to the effect on the cardiac development, but nobody has really tested it the other way, right? Like what other impacts it might have. Like secondary impacts on the embryo that might lead to the lethality. And in these NKX 2.5 knockouts, you can see that let me just start the laser pointer. Okay, you can see that because the looping process is interrupted in the beginning, instead of having a right ventricle and a left ventricle, now it's a single chambered heart. And this is something that was found out very early in the time, 1999, and these are some of the scanning electron micrographs that you can see. Plus plus means wild type, of course. So these, the first two pictures, you can see that the looping process is happening, at different stages: ventricle, atria, right ventricle, left ventricle, the outflow tract that we talked about, um, and in the mutants, you can see that the atria and the ventricle, they are pretty much the same chamber. And this is what it develops into. So this is something that I just wanted to show. I thought it was pretty cool that how a single transcription factor, when it has such importance during the development, when interrupted or depleted, can completely screw up the entire morphology of the heart and causes lethality. And with that, basically, I wanted to just summarize what we went over in last lecture. And this is actually where I should have stopped last time, but because of time, we had to stop earlier than that. What I wanted to emphasize basically throughout the first lecture, I wanted to make sure that everyone understands, just like any other organ, heart is an extremely complex organ, and the development of which is again involving a lot of these steps, these cellular events. as listed on the screen, you can see. And these are basically migration of the cardiac precursor cells, right, from the epiblast. They start migrating laterally and they build the heart fields. Along the line, they also proliferate and expand the cardiac precursor cells. Then based on the molecular pathway, some of which we discussed, such as the Wnt, BMP, and FGF, these factors, they specify these precursor cells, and eventually it leads to the growth and morphogenesis of the organ, and finally differentiation of these precursor cells into certain various cardiac structures like the ventricles, atria, outflow tract, so on and so forth. So these are some of the events associated with the development of the heart. But now we will move on to, today we will be learning a little bit about blood vessels, blood cells. And this is important because this is a segue to learning about how the coronary vessels form. So in order to understand what sort of blood vessels are found in the heart itself and how they develop, I felt this segment will be important for us to understand first how in general the blood vessels are forming and the blood cells are being generated, and then we will move into specifically looking into the heart. So I hope everyone remembers this picture, the coronal section of the embryonic disc. And I kept mentioning again and again that the lateral plate mesoderm, the splanchnic lateral plate mesoderm basically gives rise to all these circulatory system parts, which is the heart, blood vessel, blood cells of the circulatory system. This is something important to remember. So basically the portion from which the heart develops is the same portion that gives rise to the blood vessels and the blood cells. And interestingly enough, even though heart is an organ that is responsible of, pumping the blood to the rest of the embryo and organs, but the blood vessels actually, they form independent of the heart. So these two developmental processes are independent of each other. And later on, once both of them have gone to a certain extent of development, later on they connect, they link to each other. Now, this is something that people don't completely understand why this is so. Why cannot the blood vessels why can't the blood vessels just originate from the heart as the heart is developing, and then it just supplies to the entire embryo? Why does not that happen? Why do the blood vessels need to originate separately at different sources, at different in different spaces of the embryo, and then why is there a later linkage of these two events? So this is something that I just wanted to put it out there as a food for thought. And if anybody has any thoughts about it, I'm happy to discuss. But what I basically wanted you to do is maybe and this is actually a question for you, Varsha. How do folks submit, like a little essay or assignment? What would be the way to do about it?
So there is Moodle, and if you can give me the question, I can upload it on Moodle, and there is provision for students to upload the assignments.
Got it. So, okay, thanks, Varsha. So this is something that, there is no right or wrong answer here because this is an open question, and I would like for everyone to, put their thoughts, and I will, frame the question, and with Varsha's help, I'll put it on Moodle. But this is something that I would like you guys to, give some more thought, think about the timeline of heart development and the blood vessel development, and see why these two have to develop independently, and why can't the blood vessels just originate from the heart and supply to the embryo, the rest of the embryo. So this is a question that's out there. But moving on, so the blood vessels in the embryo during development, it forms by two processes basically. One is vasculogenesis and one is angiogenesis. I'm very sure all of us, we have come across these terms time and again. But just to make sure everyone is on the same page, vasculogenesis is the de novo formation of blood vessels from progenitor cells, meaning this is something that starts from scratch, right? There is no pre-existing plexus or a network of blood vessels. There is absolutely, you start from zero. So there is a bunch of precursor cells that will undergo certain steps, or experience certain molecular signals, and they will build the blood vessels from scratch. That's what de novo formation means. And it starts again at the lateral plate mesoderm. And in this picture here, I'm just telling you in a very simple manner what is happening. The lateral plate mesoderm cells, they give rise to hemangioblasts, which is a bipotential progenitor cell type. And these progenitor cells are capable of either giving rise to hematopoietic cells or endothelial cells. And hematopoietic cells will eventually go on and make the blood cells, the RBCs, WBCs, and all sorts of immune cells. And the endothelial cells will go on making the architecture, the architecture of the blood vessel, something that we are going to see now. And all this, again, BMP and Wnt, these are very key factor. As I keep saying, that mesodermal cells are always under the influence of these two factors, and these two factors are driving this process. Now coming to the sites at which, the places where vasculogenesis occurs, one is extra embryonic site, and we call it extra embryonic vasculogenesis. And the second one is embryonic vasculogenesis, which occurs from the name itself. You might be able to guess that one occurs in the yolk sac, right? The extra embryonic one. And then the one that is intraembryonic, that is occurring within the embryo itself. Okay, so let's talk about extra embryonic vasculogenesis here. It, of course, as the name itself suggests, it is outside the embryo, and in this case, it is the yolk sac. So the blood vessels of the yolk sacs are formed with this process. And these vessels, after their formation, they go on connecting to the umbilical vein of the mother for the nutrient and gas exchange. So the way it occurs is shown in this picture down here, where the undifferentiated mesenchymal cells of the yolk sac, they come together making the blood islands. And these are nothing but the precursor cells for primitive blood cells. Okay, and since they are sticking to each other, like you can see in these pictures, they appear as tiny islands in the midst of all these mesenchymal cells, hence the name blood islands. And these mesenchymal cells, they give rise to blood islands and also rearrange themselves and differentiate into the endothelial cells of the blood vessels. And soon these two cell types, the primitive blood cells and the endothelial cells of the blood vessels, they arrange in a way that the blood cells are trapped within the layer of endothelial cells, as you can see. And that is how the development of blood vessel starts, through vasculogenesis in the yolk sac. And this is a picture of a human blood island. You can see, I hope you can, just try and remember this. The blood islands are, stuck inside the endothelial cell layer. And this is a picture taken from, a yolk sac of a human embryo. And you can see the mesenchymal cells here, the yolk sac endoderm, as we saw in the last picture, and the cells, the blood island cells that are here. Now, the intraembryonic vasculogenesis, which we'll be studying in detail, especially for the organ like heart in later slides, the concept of intraembryonic vasculogenesis is that it occurs, of course, within the embryo, inside the embryo, and at different sites. One part of this sort of vasculogenesis is that it makes the dorsal aorta, right? And then the other pathway is for the embryo to build all these capillary networks from the mesodermal cells in each and every organ. And then later on, the network that these organs build, they are connected to the dorsal aorta. So there are different layers of formation happening here. There is not a single cellular source that just gives rise to all the blood vessels in the embryo. So the dorsal aorta is forming independent of all these capillary networks in each organ. And then eventually they come together and make connection and form the plexus. So the result of vasculogenesis in the embryo, the preliminary result is this. There is a bunch of hemangioblasts or angioblasts, you can call it. These bunch of cells, they proliferate and differentiate, and through vasculogenesis, they make these primitive vascular plexus. Okay, so basically there is no pre-existing platform for them to make these, tube-like structures. Blood vessels are basically bunch of tubes interconnected to each other, right? So they started from scratch, and hence the name, vasculogenesis, which is de novo formation of the whole vasculature. Now, the second method that by which these the primitive vascular plexus expands, if I may say, is angiogenesis. This is a term that is well known. It's out there in context of many diseases, or even in development, in context of developmental biology as well. So this basically means remodeling of pre-existing vascular network, and a lot of times it is associated with migration and proliferation of endothelial cells. Okay, so once the preliminary network of the vasculature is built through vasculogenesis, it undergoes angiogenesis by angiogenesis of the endothelial cells, and then later on it remodels into capillaries, arteries, and veins, which are the different kinds of blood vessels basically that carry oxygen and nutrients, and they have their own function. And this process of angiogenesis involves, again, a lot of cellular events. And I'm just showing some of these events here, such as sprouting and intussusception, which is basically migration of the migrating front of endothelial cells, and the branching. Here are the pictures on the right. You can see what that branching process means from a single vessel. So if hypothetically, if vasculogenesis creates a single vessel, then it can undergo different sort of, different kind of remodeling process, such as this branching that is happening here, and create more vessels. Similarly, if this is the primitive vessel that was formed, it can undergo sprouting angiogenesis and make more vessels, right? Proliferation, we all know. Fusion is something where if there are two vessels, they will come together and they will make one large vessel. And regression is something where if there is a vessel, a pre-existing vessel that is going to regress and disappear. Okay, so these are all part of remodeling that happens under angiogenesis. Okay, there is differentiation, transdifferentiation, where a progenitor or a precursor cell can give rise to two different cells, right? So that is differentiation. But if the two cells can convert, under the influence of certain molecular factors, can convert to each other, or start expressing markers that belongs to a different kind of differentiated cell, that would be transdifferentiation. And then finally, there is recruitment of perivascular cells. So perivascular cells are the cell types such as pericytes or smooth muscle cells. These are the mural cell types that cover the endothelial layer of the blood vessel. So a blood vessel is basically a tube-like structure. In red here, you see the endothelial cells, right? And they form a tube. And these perivascular cells are basically at the periphery of these blood vessels. They are wrapping these endothelial cells from outside. So these could be pericytes, as you can see here in yellow, or these could be smooth muscle cells. So pericytes are considered to be the precursor cells for smooth muscle cells in some scenarios, not all. And smooth muscle cells, when they wrap the endothelial tube structure, they make it more stable, and they basically, then it is considered as a mature vessel. So this is how the whole angiogenesis or remodeling process happens post vasculogenesis. Okay, so we saw that vasculogenesis was a bunch of angioblast cells giving rise to a primitive vascular plexus that is not differentiated into the different blood vessels yet. But this primitive vascular plexus, or the cells within these primitive vascular plexus, can undergo angiogenesis, and then they make the different structures through all these processes that we talked about just now: sprouting, branching, fusion, regression, differentiation, recruitment of perivascular cells. So with these processes happening, some of them happening at the same time, the structures such as veins, capillaries, and arteries are formed, and then they are connected. Okay. So now this is just bringing the vasculogenesis and angiogenesis together in terms of the molecules or the signals that are involved. So as I just said, the mesoderm cells, the mesodermal cells from the lateral plate mesoderm, they can differentiate into hemangioblasts. These are the bipotential cells that can give rise to either endothelial cells or the hematopoietic cell. We will talk about the hematopoietic cells in a bit. But speaking of endothelial cells, this is the kind of structure they make. So this is vasculogenesis. So anything in the left-hand side that you see is vasculogenesis. And this process is controlled by FGF, which maintains the mesodermal cells. And the vasculogenesis cannot happen if you deplete the vascular endothelial growth factor and its two receptors, VEGFR1 and VEGFR2. Okay, so these two molecules are key for vasculogenesis. Once vasculogenesis occurs, then there is, of course, the remodeling that is happening, and angiopoietin and Tie2, angiopoietin is the ligand and Tie2 is the receptor. This interaction is essential to maintain the capillaries or the initial blood vessels that are formed within the organ. They undergo remodeling and then formation of more mature or stable structures such as the arteries and veins under the influence of VEGF and PDGF and TGF-beta. So these are some of the key factors that are involved during vasculogenesis, which is here in the left, and angiogenesis, which is here in the right. And I just wanted to show some, some consequences of, messing up with this molecule Tie2, which is a receptor for angiopoietin. And this is in a mouse embryo. And you can see that there is hemorrhage, and there is the hemorrhage, and the liver size is quite small in these organs. And the primary reason for the lethality of these Tie2 knockout mice is the hemorrhage, basically. So it's a vascular phenotype that kills these mouse embryos. Similarly, it is not just important for this signaling between angiopoietin and Tie2 is not just important for, how to say it, not just important for building the arteries and veins, but also the lymphatics. And here is one example, and you can clearly see the difference between the control and the embryo, where the Tie2 receptor has been depleted from the lymphatic vessels. And this is how it looks. This is the embryonic skin, and there is a clear lack of PROX1, which is a marker for lymphatics. And coming to the ligand, so we were talking about the receptor Tie2, right? And this is showing what the ligand, what effect does the ligand have on these vascular development events. So on left, you see the control. The second picture is angiopoietin 1. Third picture is angiopoietin 2. And then the fourth picture is a double knockout. So even though these two, the single knockouts look okay, in the double knockout, you can clearly see that there is skin edema, which is basically the fluid is being filled under the skin, under the subcutaneous layer. So basically from this, we can, have a sense of how important Tie2 is. And people in the field, they use this ligand receptor interaction a lot in order to understand more about how the vasculature develops and how does it remodel. Coming to another major factor, which is the vascular endothelial growth factor here. I'm showing you a yolk sac, mouse yolk sac, wild type on the left and the mutant on the right. You can see the lack of blood vessels, even though I don't think this is like the best picture out there. I'm sure there are way better pictures out there, but at least you can see the vessels in the wild type here, but the lack of vessels in the mutant. So all these, molecular pathways have been validated in mice, and a lot of them have been validated in zebrafish as well, which is another cool model. Okay, so moving on, I wanted to just show this, briefly go over how the blood cells are originating. And I hope everyone remembers this lineage tree that I have been showing time and again in last lecture, where the mesodermal cells, when they start expressing MESP1, can get, they get a cardiac fate. And all of them then go on to make all these cell types that contribute to building the heart. But if they start expressing VEGFR2, which is something that can lead to the hematopoietic lineage and the vascular lineage. So let's talk a little bit about this, what happens here. So these multi-potential, these these guys here, the multipotential hematopoietic stem cells, they are, I don't know if we should say multipotential, they are for sure bipotential. A bunch of them can, they have the potential to give rise to endothelial cells, and the others can give rise to, um, um, they can give rise to the hematopoietic cells. So here you can see the flow chart of how this division occurs. The blood stem cells, they can either form the lymphoid stem cell or the myeloid stem cell, which then move on to making the myeloid stem cell can make the red blood cells, platelets, and then the white blood cells. The lymphoid stem cells can give rise to lymphoblasts that eventually gives rise to B lymphocyte, T lymphocyte, and natural killer cells, right? So this is how it happens. And I think this lineage tree is very key for everyone to understand and appreciate that it all arises from the lateral plate mesoderm cells, but based on what kind of molecule they express or are under the influence of, they get either a cardiac fate or they get a hematopoietic fate. Okay, so yeah, this is, I guess, just the summary that we talked a little bit about vasculogenesis, how this process is basically making blood vessels, a primitive plexus of blood vessels from scratch, which we also call as the de novo formation, and angiogenesis, which is followed, which follows vasculogenesis. So this involves remodeling of the pre-existing vascular plexus. Now, the cellular processes, some of which we talked about, the branching, fusion, regression, sprouting, etc., etc. The molecular drivers such as angiopoietin Tie2 interaction, VEGF and VEGFR2 or VEGFR3 interaction, and the role of, I briefly mentioned PDGF, which is involved in the recruitment of the perivascular cells and making the primitive blood vessels stabler, and helps in differentiating them into either arteries, capillaries, or veins. We looked at some of the phenotypes, consequence of signal depletion, and briefly learned about the origin of blood cells, right? Okay, so now since we know a little bit about how the blood vessels are forming and how the blood cells are forming, I thought this will be like a good time to talk about how do the blood vessels of the heart form, right? And these blood vessels that originate from the aorta and supply to the heart muscle are called coronary vessels, and these are going to supply they will form at the outer surface, and they will slowly penetrate and branch out and supply the blood to different parts of the heart and intramuscularly as well. So that is something I want to talk about. Before I move on, did anyone have any questions? I thought it was pretty straightforward, but if anybody has any questions about any of the stuff that we discussed, feel free to stop me in between. Okay. So yeah, I just said what coronaries are, right? So these are the blood vessels that supply the heart. And this is a cartoon of a mouse heart, the dorsal view of it. And people have studied the coronary vessel development in mice, if not extensively, to a great extent. And as I keep saying, zebrafish is also a model worth looking at. So at your spare time, if you want to, today I'm going to speak majorly about mice and how the coronaries develop in mice. But if you are interested, you should also look up zebrafish heart development and how the blood vessels there develop. And zebrafish is a much, how to say, handy uh, model to work with, because of course you can do live cell imaging there, and the genetics is just so how to say uh, uh, less time consuming, if I may say. And the main thing is that it allows for live cell imaging. So you can watch all the cellular and molecular events through time lapse imaging. In mice, usually we have to do time course imaging, right? So that is something worth looking at. If you are interested, if you have time, please make sure to look at it. So within the heart, the vasculature develops through intraembryonic vasculogenesis, followed by angiogenesis, right? So anywhere within the embryo, take whichever organ you want, the first step to build blood vessels is vasculogenesis. You have to first build the preliminary plexus that you see here. The organ first needs to build this through vasculogenesis. And then later on, through the process of remodeling, you can make these blood vessels. Okay. So in the mouse embryo, here you can see this preliminary plexus. The vessels in this plexus, they come together, they coalesce and make and start expressing the certain markers that either make them venous or make them arterial. Then they connect to the aorta, which is the primary source of blood. Once they attach, the blood flows through them. And very interestingly, the blood flow initiates or triggers the remodeling process of these coronary arteries. So once the stem, they called it a stem, once the stem is formed and blood starts flowing through these vessels, the remodeling is fast tracked, and you can see that it is not only just growing in length and in width, but it is also branching and expanding throughout the organ. And this is something that we are going to look at step by step, what happens every step of the way. Talking about the first step, okay? So how does this initial plexus of the heart is being made? And it's called the coronary plexus. So there are basically two sources that give rise to the vessels in the heart in order to make that preliminary plexus. Now, the first source is sinus venosus, which is the venous inlet of the embryonic heart. And I will show you a picture of that in a bit. And then the second source is the endocardial endothelial cells. If you remember, I showed you this picture, and this is a sagittal section. If you take this heart and you make a sagittal section, right, just cut it open, you will see that this is the ventricular chamber, basically, in a wild type heart. And that's the muscle part, the muscle of the ventricle. Now, the cells that are lining this ventricular lumen, they are covered with a smooth cell type, which is called the endocardial endothelial cell. So these cells are only found in the lining of the ventricular chamber, okay? And nowhere else, basically. So the second source is that endocardial endothelial cells, and the first source is the sinus venosus cells. Now, these two cell types, they either through expansion and migration or migration, they give rise to two independent plexus of cells which come together and make this. Okay. Okay, this is a busy slide. but let's go through it one by one. So remember the first, on the top panel, I'm showing the first source of these, the first source, which is the sinus venosus, for making these preliminary vascular plexus, which is the sinus venosus. Now the sinus venosus is present here, okay, which is marked by a marker conjugated to lag Z here. And the second source is the endocardium, which, as I showed you here, is inside, right? It's you cannot see it from outside. So this is very deep section of the developing heart, and you can see it inside, and you can see that it is lining the ventricle. Stop me if you cannot understand, and I will try to show some other pictures if that helps. But let me go through this first. So these are the two sources basically. Now let's talk about sinus venosus, which is the venous outlet, and the arrows show you the direction of migration of these endothelial cells. You can see the dorsal. This is the dorsal view of the heart, and you can see that it is migrating downward, right? And it is covering the. It is starting to cover the entire dorsal part of the heart. And finally, it starts rearranging. And I don't know if you can see, there is a dark, vessel here and more clear vessel here. And then they start rearranging. After they form the plexus, they start rearranging, making the coronary arteries and veins, okay? And this is called outside in, because they first cover the outside of the heart, and then slowly they start penetrating in. So wherever you see the bold or the filled in arrows, that is the migration pattern outside. But once you see these empty arrows, that means the vessels are now going inside. Okay, so it's called the outside in pattern. It starts to cover the sinus venosus endothelial cells. They cover the outside first, and then they slowly go inside, so outside in. Now coming to the endocardium, endocardial endothelial cells, they are lining the ventricle, as you can see here, and they go inside out, okay? So you can see these empty vessels, they started here, and now they are going towards outside. So there are, you can see that they have now started filling up the entire heart, and some of these blue that is at the periphery is also coming from the sinus venosus. So it's hard to distinguish, but hopefully in the few next slides you can distinguish that. So this outside in and inside out pattern, right? These two independent sources, they make their own plexus, and then they eventually come together and fill up the entire heart, as you can see. And then they make the coronary arteries and veins. So this is a cartoon of what I was trying to show in actual data, which I'm not sure if you guys got. If you have any questions, stop me, please. So this is the dorsal part of the mouse heart, dorsal view of the mouse heart. The sinus venosus, as I was saying, is this structure. This is the venous inlet of the embryonic mouse heart. And the surface vessels, they grow in this direction, which are showed in blue, right? And as they grow, they will fill up the dorsal side, and then they will start penetrating the ventral side also. So they will grow like this, and then this is the ventral view. They will come from behind. If you're looking at the ventral view, they will come from behind also. Now the endocardial cells, which you can see here in pink, they come from inside, right? They line the ventricular lumen. So they come from inside and they start moving outside. So there are two, both migrate and proliferate as they make their own plexus, which will be either in blue or in red. But the fact remains that other than the direction, the rest of the processes are mostly same. It's basically they are migrating and proliferating as the developmental time proceeds. Okay. So I wanted to, yeah, this is basically the cross section of one of these hearts, right? So the endocardium will be lining these ventricular chambers, and it will start its migration somewhere here, and it will proceed intramyocardially, like piercing through the ventricle, and it will reach the surface. However, the sinus venosus, it will start from the outside and slowly it will penetrate and it will meet the endocardial plexus somewhere in the middle. So endocardial plexus is inside out formation, and the sinus venosus plexus is outside in formation. Did I make sense to everyone?
Yes.
All right.
I had a question.
Okay.
Do they undergo remodeling in a synchronized manner, or their remodeling also happens independent of each other?
Well, that's a great question, actually. There is, I mean, the coronary arteries, they appear almost at the same time, whether you lineage trace the ventral, the sinus venosus, the label cells or the endocardial label cells, but the coronary arteries are appearing almost at the same time, which is something between embryonic day 14.5 or 15.5. So I would like to say that the remodeling is occurring in a synchronized manner. Else, so both of them have to somehow communicate with each other, right, in order to make these coronary vessels. If the communication is impacted, this vessel development is delayed. So I would say that they are synchronized.
Okay, thank you.
Yeah, even though it is not explicitly showed in an experiment that was done in mouse embryos, but that is just like a logical explanation that I can come up with. Okay. Okay, just to show you some very cool confocal images, very crisp images here. I'm just trying to I'm just making an effort that everyone gets this inside out and outside in. So this is the sinus venosus plexus that I'm trying to show. And hopefully these images, they do justice to what I'm trying to say here. The sinus venosus is labeled here using this Cree line in mice. You can see the SV means sinus venosus. It is labeled here, and as time passes, right? This is embryonic day 10.5, 11.5, 12.5. You can see that they start migrating. So this is definitely like not the same heart, because in mice you cannot do live imaging, or there are no ways, at least as of yet, where you can open the chest and look at the same mouse heart undergoing these kind of events. So these are like different hearts at different time points, but the labeling is the same. So all hearts were labeled, all the sinus venosus cells in these hearts were labeled, and then they followed the pattern through time course imaging, and this is what they find, that the migration is happening. And hopefully everyone can now relate to this cartoon when I say that there is this surface migration, which is shown in blue. And eventually these vessels will, after covering the heart, they will also start penetrating deeper into the muscle, so that they can meet the endocardial cells halfway, right? And this is the other ventral view that I was trying to show. So I'm just trying to show that the endocardial cells, which are in this case labeled with NFAT-C, one Cree, which is another marker for, it's a Cree line that labels the endocardial endothelial cells. So they origin in the middle, right? As you can see here, because they are lining the lumen of the ventricle, and they migrate out. So this is inside out, right? This is outside in, this is inside out, and eventually they meet together and they make that plexus. So yeah, it's a little tricky to understand with like these 2D images, but I hope I made my point, that the initial plexus that is formed, the initial capillary plexus that is formed has two cellular sources, the sinus venosus and the endocardium endothelial cells. And once this plexus is formed, a certain group of cells that are in proximity of the aorta, they go on and attach to endothelial cells of the aorta. Okay, so they make a little vessel and then they go attach. And after that attachment, because aorta is the primary blood source, right? So the blood can now flow through these arteries and the remodeling process starts. Now, I just want you to know that this attachment is called as a coronary stem. Okay, so this process is called stem formation. And this has been shown through, again, time course imaging. This is the aortic lumen, right? And below every picture, you can see a cartoon. So hopefully that helps. This is the aorta. This is the right coronary artery site. Okay. And as time progresses, as it is embryonic day 12.5, you can see that some vessels that were formed in the primary, the preliminary plexus, these vessels are now connecting. These vessels are in blue, basically, are now connecting through this red attachment. These are connections with the, with the aortic endothelial cells. Sorry about that. So once that connection happens, it's called a stem. This is a stem that connects the coronary vessels with the aortic endothelial cells. And now the blood will start flowing, right? So you can see here, this is the stem that connects the coronary vessels CV to the endothelial cells of the aorta. And now it starts remodeling and it will get blood flow. And the minute it gets blood flow, which is embryonic day 13.5, it sets on onset of flow. It will acquire some smooth muscles and it will be stabilized. And once the blood flow goes through these arteries, it will start remodeling. Okay. So this is basically, this is basically showing that, and the picture quality is not as great as I thought, that this vascular plexus, it undergoes remodeling upon the onset of blood flow. And once this blood flow has started flowing through these tiny vessels, right? Blood flow is blue in color here. Then the arteries and the veins, these mature vessels are formed. And this is something that you can see in these high mag pictures of the mouse heart. The low mag pictures are not so clear. But right after the blood flow is received, these vessels are undergoing remodeling and they are maturing. And you can see that they are more, distinct from the rest of the vessels. Okay. Okay. So as I said, plexus formation, then a group of cells, they attach with the aortic endothelial cells, and that attachment is called stem that allows blood to flow through them. Then the remodeling happens, right? The vessels are growing in length, in width, they're branching, they're recruiting these yellow tiny pericytes and smooth muscle cells around them. And then finally, the final step is the smooth muscle or the pericyte recruitment. These are, again, if you remember, are the perivascular cells, okay? And this is what, how the mouse heart looks over the, over the developmental period. This is E13. I think this is E13.5. basically when the blood flow is initiated, because there is this stem formation between those two endothelial cells. Once it gets blood, you can see that these vessels, so these are the endothelial cells, okay? The bottom panel are VE cadherin, which is a marker for coronary endothelial cells, which is also shown here in gray. And you can see that once the blood flows through these vessels, they start remodeling here. They are coming together and they make these arteries, right? And as they are making these structures, the smooth muscles are also recruited. So somehow, until the vessels get blood flow at E13.5, the smooth muscles are not recruited. But once after 13.5, when the blood is flowing through them, these smooth muscles are recruited, and they are making these vessels, more stable, which allows a higher volume of blood to flow through them. And this red is smooth muscle myosin heavy chain, which is a marker for the smooth muscle cells. Any questions? Okay. Okay, so for vasculogenesis and angiogenesis, we learned about VEGF signaling, the Tie2 signaling, and here I just wanted to add the jagged1 and notch signaling, which is very key for this process, the smooth muscle recruitment process. Jagged1 is expressed in the endothelial cells and notch is expressed in the smooth muscle cells. So this is how the recruitment happens. Once the endothelial, the ligand is in the endothelial cells, which helps recruit the smooth muscle cells or the pericytes onto the endothelial cells of the blood vessels. And these pericytes and smooth muscle cells are expressing notch. So that signaling between the endothelial cells and the smooth muscle cells is mediated by jagged and notch signaling. Okay, so this is like a extra layer of regulation that happens during the development of coronary arteries. So I hope this, this cartoon now makes a little more sense than it did before. The plexus, the initial plexus is, it's created by sinus venosus and endocardial endothelial cells. It's called the coronary plexus. Then few cells of the coronary plexus, they connect to the aortic endothelial cells, and they make this stem that allows this attachment or stem formation allows the blood to flow through them. The blood flow remodels the coronary artery, expansion, and it is elongating, it is getting wider, it is branching. The smooth muscles and pericytes are being recruited, and that again makes the vessel more stable, and that allows for more blood to flow through them. And this is like the final product, coronary artery, after the smooth muscle and pericyte recruitment, right? Okay, now talking about, I just want to briefly mention some, molecular pathways that are key. By now, most of you must already, you have realized that vascular endothelial growth factor is one of the key factors. The name itself tells us that it is one of the vascular factors that is responsible for maintaining the vasculature and pushing the vasculature in all directions for remodeling processes. And VEGF-A, vascular endothelial growth factor A, and its receptor, this R stands for receptor 2. This interaction is the most important interaction during the development of the vasculature. And this basically regulates two main cellular events, which is migration of endothelial cells and proliferation of endothelial cells. These are the two key events that happen. Here is the same picture where sinus venosus is in, this is the dorsal view of the heart, this is the ventral view. Sinus venosus is giving rise to all these superficial vessels that eventually will go deeper. And when you flip the heart, you get the ventral view, and the endocardial vessels are, giving rise to another a secondary plexus that will connect with this, the plexus that is being created by sinus venosus here. And each of them, each plexus is regulated by two different set of, programs, developmental programs, basically. Talking about sinus venosus giving rise to its plexus, first one is VEGF-C, right? Which is expressed by the epicardium of these mouse hearts or embryonic hearts. And the sinus venosus cells, endothelial cells, are expressing VEGF-R2. So there is your receptor, and the blue part will have your ligand, and which is how the migration is driven from one source towards the other. Okay. Similarly, Ella-Bella is one of the sources that is found in this blue part throughout the surface of the heart. And APJ is the receptor that is found in the sinus venosus. And since the receptors are residing in the sinus venosus and the ligands are expressed elsewhere, the receptor expressing cells are attracted towards the ligand expressing cells, and that is how the migration is happening. Okay, I hope that makes sense. Similarly, when we talk about the endocardial cells, there are two molecular pathways that are very key. One is the VEGF-A, VEGF-R2, as I was talking here, and the another one is CXCL12 and CXCR4. So in both cases, similarly, the receptors in CXCL12, CXCR4 pathway, the receptor is CXCR4, right? And the ligand is CXCL12. And in the endocardial plexus formation, the receptors are expressed by the endocardial endothelial cells, and the ligands are expressed by the myocardial cells that I showed you earlier, that the compact myocardium and the trabecular myocardium that build the ventricular wall. Right? So since the ligand are here and the receptor is here, right? The ligand is in these muscular walls and the receptor is lining the ventricular chamber. Hence the direction is in this way. Okay. So I hope that makes sense, that the receptors are always almost expressed by the endothelial cells, either of the sinus venosus or the endocardium, and the ligands are present either, in this case, it's present in epicardium, hence the migration is superficial. And in this case, it is present in intramyocardially, hence the whole migration and proliferation process is more deeper. And now this superficial plexus will now combine with this, plexus that is formed little deep, and they will come together to make this whole, a complete wholesome plexus for the heart. Okay, yeah, this is, I just wanted to mention that VEGF-A and VEGF-R2 expression of both the ligand and the receptor are hypoxia inducible. And some of you must already know it in context of cancer, because cancer angiogenesis is also very, it's been studied a lot in context of hypoxia. And here you can, this is just a picture showing that when the conditions are normoxic, which is, optimal oxygen is available for the cells, there is restricted growth, the right amount of growth. But when there is higher hypoxia, meaning lower oxygen, the concentration of VEGF goes up. And that's when VEGF-R2, which is the receptor for VEGF, and is expressed in the endothelial cells of the blood vessels, starts growing, in this case, a lot more than what it would in normoxic conditions. And there are, all these things happening under hypoxic conditions that facilitate this excessive growth. Okay, and this is important in context of development because in the uterus, when the embryo is just sitting in the uterus, it is a hypoxic condition, right? So because of that slight hypoxia, these molecules are playing a great role in initiating and regulating and making sure that the angiogenesis process is happening in the embryo. So hypoxia is not necessarily a bad thing always. In context of cancer, yes, it is a bad thing probably, but during development, you need it at the right time, at the right spot for the organs to develop correctly. And in this case, it regulates the vasculature development. Okay, hope that makes sense. Okay, VEGF, the vascular endothelial growth factor, also controls the non-migrating front. Here we show that, I showed that in response to this VEGF, the migrating front, right? The cells that are at the migrating front exposed to the ligand are migrating excessively or furiously towards the ligand. But what happens in the non-migrating front is also to some extent controlled by VEGF. And how is that basically? I'm sure some of you must have heard of tip cell and stalk cell concept, and this is mostly studied in context of retinal vasculature development. In this case, the tip cell is at the migrating front. As you can see, it is at the front, which is going to sprout out, right? So this is the base of the vasculature, which is connected to the stalk, and the tip cell is sticking out, trying to sense what is out there, what kind of molecules are out there. It senses that, and then it decides whether it needs to migrate out or whether it needs to retract and migrate in a different direction, okay? And in green here, you see what is happening in the tip cell, and in blue here, you can see what is happening in the stalk cell. So when there is hypoxia, right, when the oxygen content is suboptimal, basically, the expression of VEGF-A is induced by cells surrounding the vasculature. And by doing so, the VEGF-R2, which is expressed in these, vascular cells, it senses that, okay, there is ligand out there, and it decides that it needs to migrate. The cell decides that, okay, I need to migrate out. But at the same time, expression of VEGF-R2 also leads to DLL4, which is the ligand for Notch signaling. And if this ligand is expressed and is produced, it will interact with the Notch, which is the receptor, right, in the stalk cell, and it inhibits the VEGF-R2 expression in the stalk cell. So this is how now this combination of molecules, expression of VEGF-R2 and the Notch ligand, DLL4, this combination helps the cell decide that, okay, I need to sprout, and this combination lowering the activity of VEGF-R2 and enhancing the activity of Notch makes the cell non-sprouting, basically. So it knows that, okay, I need to just proliferate and not sprout out. Because if every single cell will sprout out, it will be a mess, right? So this is how it's been decided which cell becomes tip cell and how it responds, when it responds, and which cell is the supporting stalk cell. Okay, and here is a picture from the retina, from the eye. You can see that all these arrows, the triangular arrows, these are all the tip cells, and all the asterisks or the stars here are the stalk cells. And all these tip cells, they have these pilopodias that are, they are thought to sense. They are thought to be mechanical sensors and sensors for other, molecules out there. And these are the supporting stalk cells. So there is a complex, molecular interaction between VEGF and Notch that decides which direction to proceed, how much to proceed, when to sprout, so on and so forth. Okay, so VEGF not only controls the migrating front, but it also specifies the non-migrating front, which is the stalk cell here. VEGF-A and VEGF-R2 interaction is also known to help proliferation, and I'm not showing any in vivo data here, but this is basically folks have taken the ventricle, they have dissected out the ventricular chamber of the heart, and they have tried to culture it, right? So in this case, the blue portion will be all endothelial cells. It's basically the lumen that is there, and then the red portion is the muscle part, right? And you can see that in the control hearts, control ventricles, you can see a lot of endothelial cells. And in the hearts where they have treated it with SU1498, which is a drug that inhibits this signaling, the number of endothelial cells goes down. And I'm not sure if I'm showing it here, but this also correlates with one of the proliferation markers. When they add BRDU or EDU, I believe it was EDU, they could see that fewer endothelial cells were proliferating. So the point that I'm trying to make here is vascular endothelial growth factor does many things, including migration and proliferation for the endothelial cells. And it is one of the key molecular factors that regulate development of coronary vessels in the embryonic heart. Okay, so yeah, I think we are, what's the time? Oh, we still have time. So this is basically the summary of what I just spoke. We talked about the sequence and timeline of coronary development, right? How the initial plexus is formed from sinus venosus and endocardium, how the stem is created, how those two endothelial cells of aorta and coronary vessels attach, get the blood flow, and then the remodeling happens, and then they become stabler by recruitment of neural cells. And what is the role of VEGF in all this? Not in all of it. We discussed some situations where VEGF plays a very critical role, but there are also other pathways that I did not discuss in much detail, such as the CXCL12, it is a chemokine, PDGF, and Notch. Notch we talked a little bit about, which also play a very crucial role in development of vasculature in the heart. Okay. So yeah, this is the summary for both the lectures, I guess. So there were 3 parts. I hope everyone remembers that in part 1, we talked about how the heart develops, cardiac organogenesis. In the second part and the third part that we talked about today, we talked about how the blood vessels arise through vasculogenesis and angiogenesis, and how the blood cells arise from the hemangioblast. And then we talked about the development of coronary arteries. I think that is the end of it. Yeah, and I just listed like a few references that I used for these 2 lectures, and some fun readings, articles, if somebody is interested to know the source of these images and information. So any questions for me? And I will actually communicate with Varsha and put up that tiny assignment that we talked about as to why blood vessels are originating independent of the heart. I will formulate it and put it on Moodle so that you guys can feel free to share your thoughts on that. So I guess that's it on, yeah, that's it. So any questions you guys may have? Okay, I'm going to start my video. Are we all good? No questions? Okay, then. Varsha, so what do we do now?